Drug repurposing is one of the most powerful tools in our toolshed to look at countering diseases like COVID-19. Look at the screen in front of you. This contains data obtained from Stitch and other similar databases like Metadata or CTD, where you have a list of validated drug protein interactions. The left column indicates that it's Homo sapiens, which is the species, and you have protein IDs, and you have a corresponding smile string and the generic drug name on the right hand side. There is no one-to-one interaction between different drugs and their proteins, although our primary interest is in the drug target that modulates the disease. Now, because there is a many-on-many relationship between the two, what we can do is we can use this validated information, filter it, clean it, and then store it in a graph database and ask the question, how many proteins does my inhibitor or drug or list of inhibitors interact with, and what kind of interactions are they, and are there any drugs in this database that have a substantial overlap in terms of the proteins they interact with more importance given or more weightage given to the current drug target, but also a substantial overlap with other proteins that my current inhibitor interacts with. This gives you a good indication of the biological fingerprint for each drug, and the assumption is that if the overlap between two drugs of interest in terms of their biological fingerprint is high, then they should also probably be considered for the disease at hand. Let us go to the next step in this. Once you obtain this information, keep it in a database. You could either directly query, as I said earlier, or you could actually program something in Python instead of querying the graph database, which might be computationally expensive, and you can group it by the list of proteins that each drug interacts with. So here you see a list or an array on the right hand side of the proteins that interact with the drug on the left hand side. Now the next step is to find the intersection divided by the union of the list of proteins that each drug interacts with all others. So take the first drug and go through the list till the end in a loop and compute the intersection divided by the union for each of those. This is just one way to quantify the similarity or the overlap between the proteins they share, but you could look at other metrics. And once you do this, you can actually define a distance or a similarity between each pair of drugs in the database based on their biological footprint. And what you come up with is a drug-drug network, a drug-drug similarity network, or a graph. Again, this graph can be stored in GraphML or a variety of other formats using software like Gephi or Cytoscape. These can be visualized, these can be interrogated, or you could store a lot of this information in a graph database and query that. But the query will always be, given my drug or drugs of interest, which are the other drugs that not only share my current drug target, but also a substantial number of other drug targets with my drug. So this is a 2D image, so you can't actually move it around, but in visualizers, you can actually move this, you can expand this, you can find your drug or drugs of interest and find the neighborhood of those, and then hypothesize that these other drugs have a similar mechanism of action and thus might be active against your disease because they share these drug targets. Now, what is different in the case of COVID is that typically this deals with a human protein network, so you'll have to be careful in also using the viral proteins to build such a graph database and to see whether you obtain enough information. But in general, if you find that a drug like is useful for treating COVID, and if it's got the same kind of interaction, there's a high chance that the other drug would also probably be active.